On Monday, we went through structure of. Did it kick me off again? It did, didn't it? Okay. Hold on. On Monday, we went through structure of membranes and some basic transport. Went through the 5 different types of transport: passive and active, diffusion, osmosis, facilitated diffusion. Okay, active transport. And today we mentioned endo and exocytosis, bulk transport. We'll go through that today. All right. Before we go through endo and exocytosis, I want to go through the last part of active transport that we cover. Okay? And active transport, before we left off on Monday, we went through this. This is active transport. Characteristics of active transport was the use of ATP, right? So after you hydrolyze and utilize ATP to move molecules or substances in some direction. Typically, active transport is against the concentration gradient. And that's what you see here, the movement of sodium ions. All right, sodium ions are moving outside against the concentration gradient. Out here it's high sodium, right, and low potassium. Okay? So we're moving sodium against its concentration gradient, moving potassium against its concentration gradient by utilizing ATP. Okay? What this does, all right, now it develops what we call a membrane potential. What do I mean by membrane potential? What happens, so here's our membrane. I'll just do it small. Here's our transporter. Right? And in every time ATP is utilized, how many sodiums are moving out? 3 sodiums. Okay? How many potassiums are moving in? 2 potassiums move in. Okay? Now, let's go through a couple different, we'll say this goes through 10 rounds of transport. All right? So now that's a total 10 rounds. How many sodiums are outside? 10 rounds? 30. All right? How many potassiums are inside? 20. All right? Do you see the difference? What has happened? How many more positive ions are there on the outside of the cell? All right? So there's a net positive charge. Okay? There's a difference. Okay? What that creates is a gradient. Okay? There are more positive ions on the outside than on the inside. It is similar to a concentration gradient. This is what we call an electrochemical gradient. All right? This gives what we call a membrane potential. All right? There's a voltage difference. More positive ions on the outside versus the inside. Okay? There's a voltage difference across the membrane, on one side of the membrane versus the other. This is collectively called an electrochemical gradient. All right? So we have the chemicals represented by ions, sodium and potassium. For sodium, there's a concentration gradient, correct? High outside, high sodium, right? Low potassium. Inside, we have high potassium, low sodium. So there's a concentration gradient for those chemicals. But associated with that as well is the charges. There are more positive ions on the outside than there are on the inside, based upon the movement of sodium and potassium, the unequal transport that's occurring. All right? What this does is produces a chemical force. Okay? That's the ion's concentration gradient, sodium and potassium want to go one way, and the electrical force, the positive ions themselves, have a concentration gradient. All right? So the potential for the ions movement. So what's going to happen when, say, a channel here, what we call a ligand-gated channel, this sodium-potassium channel opens. What's a channel? A channel is an example of what? Facilitated diffusion. So now if that channel opens, what's going to happen to sodium potassium? It's going to move according to its what? Its electrochemical gradient. Sodium's going to move in based upon its concentration gradient. Potassium's going to move out until it reaches equilibrium. And they're going to reach equilibrium both on the ions, the electrochemical or the charge, and on the chemical gradient for sodium and potassium. Okay? Now that channel closes, what does this? Then this takes over again. The channel closes, and then this type of transport takes over to help recreate the membrane potential. Okay? So in certain cells, this is important. What cells is this important for? Emily? Muscles. All right? For muscles, there's a membrane potential which then allows for a nerve impulse to come down, right? You get a transfer of neurotransmitter from the nerve to the muscle and causes muscle contraction, right? This ATPase, or this sodium potassium pump, helps generate the membrane potential that allows muscles to contract, all right? For nerves, this helps to produce the membrane potential that allows nerves impulses to travel, okay? And we have what was called depolarization of that membrane occurs, meaning the polarized nature of it, high outside, low inside, all right, that difference of chemical charge, that depolarizes, meaning that's going to change, that channel opens, movement of chemicals in and out, and that allows what we call an action potential to be propagated down the nerve. Okay? So that's importante. All right, this is an example of an electrogenic pump. All right, electrogenic pump. It's going to pump ions across the membrane to produce an electrochemical gradient. All right, in bacteria, of this called a proton pump. All right, ATP is utilized to push protons towards one side of the membrane. Okay, so it goes from inside to outside, produces what we call a proton motive force. All that is a fancy name for a proton concentration gradient. All right? Now, what can happen? You release that channel, you can do some type of work. All right? Allows the cell to do some type of work. And this is similar to what occurs in the mitochondria during electron transport, all right, in producing ATP. Mitochondria produce this ion gradient and then open the channel, and as it comes back through, it produces ATP. An example of this is co-transport. All right? So this is a way of getting things inside the cell without utilizing, in one way, utilizing ATP. Okay? So produce the concentration gradient of protons on the outside of the cell, right, by utilizing ATP. Now, based upon diffusion principles, that proton wants to move back across the membrane, right? It wants to move back. So it wants to move from here of high concentration to low concentration. But proteins have evolved so that they utilize this, the desire of the proton to move back down its concentration gradient, and will move sucrose in with it. Co-transport just means two things are transported together. All right? In a way, the sodium-potassium pump is a co-transport. Two things are moved together, right? Sodium is moved out and potassium is moved in. They're moved in opposite directions, right? So that's called a antiporter, moving in opposite directions. This is an example of a symport protein. Sym, S-Y-M-P-O-R-T. All right? Symporter, the two chemicals or two substances you're moving are moving in the same direction. So an antiporter and a symporter. But those two substances are transported together. Questions on membrane potential and the movement of ions in and out of the cell, or symport, antiport, co-transport in general. All right, I wanted to clarify that point towards the end of the class. We didn't make that point clear that this was an important thing for producing the membrane potential of cells across the cell membrane. What did I just get there?
Your turn on your system.
Oh, thanks. I gotta shut that off. So the next part, okay? The next part of the chapter, the last part, is endo and exocytosis. So endocytosis. What does endocytosis mean? All right, so substances are taken in. So the cell takes something in. Okay, cell intake. This occurs in bulk of something big or a lot of something is internalized into the cell. All right, so it's either large or an amount, all right, or a large quantity, all right, either large in size or amount. And we have a couple different types of endocytosis. What do we know about endocytosis? Phagocytosis. We have pinocytosis. And we call receptor mediated endocytosis. All right? For the most part, you are probably familiar with phagocytosis. It is a cell engulfing some type of whatever it might be, bacteria, the best known example, right? Internalizing it into a vacuole and then doing something with the cell. So it ends up looking, here's our cell, here's our white blood cell, here's our happy, not so happy bacteria, right? Eventually, the cell will engulf the bacteria. It's a larger vacuole than it needs to be, but you get the idea. And then it becomes internalized, right, into a vacuole. Phagocytosis. Phagocytosis means something solid, right, such as a bacterium, is engulfed or brought in. All right. Pinocytosis literally means cell drinking. So the cell internalizes some type of fluid, all right, some type of liquid matter. Whether it be water in the case of amoebas that live in freshwater environments, they'll engulf water in order to produce osmoregulation, prevent osmosis. Okay? And then the last type is receptor-mediated endocytosis. And this occurs in mammalian cells quite often. All right? In receptor-mediated endocytosis, growth factors are deposited on cells, and they react with receptors. All right? When they react with receptors, those receptors are bound to the ligand, the protein that's going to shape, interact with that receptor. And that receptor then needs to be recycled. So internalized and broken down and then recycled. You'll see what that looks like in a second. All right, so here is phagocytosis. Example, your transmission electron micrograph showing a bacterium, right, which is being phagocytosed into a food vacuole. So the amoeba literally is wrapping around it, and then that will be fused with the lysosome to break down the cell. All right, break down the bacterial cell. This happens in your white blood cells. White blood cells do this all the time. Macrophages and neutrophils will phagocytose cellular debris from dead cells, bacterial microorganisms, etc. And then you have the diagram showing your food particle eventually ending up in your food vacuole. All right? Those two extensions that wrap around the cell are called pseudopodium, false feet, so they move around. What do you think is necessary from what part of the cytoskeleton is necessary for pseudopodium and phagocytosis? What element of the cytoskeleton is necessary for phagocytosis and the production of pseudopodium? Microfilaments, which means they're what protein? Actin. Okay? These are microfilaments. It's not microtubules because it's not cilia or flagella. All right? These are microfilaments. Good. Is pinocytosis, the internalization of some type of whatever's inside the or the extracellular fluid. Whatever's outside the extracellular fluid is internalized. So you see this cell just internalizing whatever's out there. Okay? You'll see this is called a coated pit. Okay? This does not happen just anywhere in the membrane. Okay? The membrane, the cell produces proteins called clathrin that line that pit. Okay? So the protein collects at an area of the cell membrane, which then targets that portion to be internalized. Then you have a coated pit, which becomes a coated vesicle and will determine what happens to that. Receptor mediated. Again, you have the coated pit, the clathrin coated pit. That's where endocytosis is going to occur. Okay? It comes in, and you end up with the receptors here within there are then recycled. Right? So from here, this will end up, this receptor will be, you'll end up with the, these receptors will be returned back to the cell membrane. So here's your receptor without the ligand, and then it gets returned back to the cell membrane. This is a way for cells to increase efficiency. The cell doesn't have to reproduce, retranslate another receptor protein. It can just reutilize it, right? Reuse it, recycle it. Okay? Questions on that? Okay? The last thing we want to talk about is exocytosis. What is exocytosis? Okay? That is movement or transport. Let's do bulk transport of proteins out of the cell. Where are these proteins likely made? The rough ER. So we're going to go from the rough ER with our studded ribosomes. Okay, then to where? You have a little, let's do that. A little vesicle that's going to come off there. Where's it going to go next? Going to go to the Golgi apparatus. Okay, So we go from ER, rough ER, to the Golgi apparatus, right? And when then finally we're going to reach the plasma membrane, where fusion is going to occur between the vesicle and the plasma membrane, and whatever's inside this will be released out of the cell. Okay, so exocytosis is just movement of bulk materials out of the cell. Questions on transport. All of these, endo and exocytosis, utilize ATP. Where do they utilize ATP? I haven't said anything about it, so how do they utilize ATP? Damn.
Together across the membranes.
Well, it's just going to, these membranes are going to fuse because they're phospholipid bilayers. So they'll just fuse without utilizing ATP. But to get it from where? Does endocytosis, pinocytosis, exocytosis just happen at anywhere in the cell just randomly? No. Does this vesicle just float and hope at some point, just float around within the cytoplasm and hope at some point it reaches the membrane? No. This is directed. Things move out. Okay? Cells that do this, such as epithelial cells, are polarized, and the membrane has to reach a certain direction. It has to go somewhere in a certain direction. So how does it do that? How does the cell know to move this in a directed motion towards the cell membrane? What does it use? Sabrina. It follows the cytoskeleton. When we talked about the cytoskeleton, we also talked about actin and myosin, and microtubules and dynein. Myosin and dynein are molecular motors. Okay? So as the cell moves these vesicles around, it is following the cytoskeleton and pulling them along. And as it pulls them along, it's utilizing ATP to direct them from the Golgi apparatus to the membrane where fusion occurs. And then the proteins or whatever's internalized in the cell can be taken care of. All right, so this is all an active process. ATP is utilized. All right, it's hydrolyzed completely. Questions on transport? On any transport? All right, on Friday, we will be moving on to the next chapter. Okay, I stopped